Positron emission tomography isotopes and radiopharmaceuticals, production, organic tracers, and synthetic challenges. Positron emission tomography, PET, is a highly sensitive molecular imaging modality that enables quantitative visualization of physiological and biochemical processes in vivo. The technique relies on short-lived positron-emitting radionuclides incorporated into biologically active molecules. This report focuses on PET isotopes and radiopharmaceuticals, including isotope production, imaging procedures, commonly used organic tracers, synthetic methodologies, and associated challenges. Particular emphasis is placed on clinically relevant radionuclides such as fluorine-18, carbon-11, gallium-68, and zirconium-89, alongside advances in radiochemistry enabling targeted imaging in oncology, neurology, and cardiology. Positron emission tomography has emerged as a cornerstone technology in modern diagnostic medicine and biomedical research due to its ability to provide functional and quantitative information at the molecular level. Unlike conventional imaging modalities that primarily depict anatomical structures, PET enables the assessment of metabolic activity, receptor binding, and biochemical pathways in living systems. This capability is made possible through the use of radiopharmaceuticals composed of a positron-emitting radionuclide chemically integrated into a biologically active molecule. The design and synthesis of such radiotracers require careful consideration of both nuclear and chemical properties to ensure compatibility with biological systems and imaging timelines. PET imaging procedure. A typical PET imaging procedure begins with the production of a positron-emitting radionuclide, most commonly via cyclotron irradiation of enriched target materials, or, in some cases, through generator systems. Following radionuclide production, rapid radiochemical synthesis is carried out to incorporate the isotope into a suitable precursor molecule, yielding the desired radiopharmaceutical. This step is followed by purification and rigorous quality control to ensure high radiochemical purity, sterility, and appropriate molar activity. The radiotracer is then administered to the patient, typically through intravenous injection. After administration, the compound distributes throughout the body according to its biochemical properties and target specificity. A defined uptake period allows the tracer to accumulate in tissues of interest, such as metabolically active tumors or receptor-rich regions of the brain. Imaging is performed using a PET scanner that detects pairs of 511 kiloelectron-volt gamma photons generated by positron electron annihilation events. These coincident photons are emitted in opposite directions and are detected simultaneously, enabling three-dimensional reconstruction of tracer distribution. Quantitative analysis is subsequently performed using parameters such as standardized uptake values, which reflect the relative concentration of tracer in specific tissues and provide clinically relevant insights into disease processes. PET isotopes, production and decay characteristics. PET imaging relies on radionuclides that decay via positron emission, each characterized by a specific half-life that dictates its suitability for particular applications. Fluorine-18, with a half-life of approximately 109.7 minutes, is among the most widely used isotopes due to its optimal balance between decay time and synthetic feasibility. It is typically produced via proton irradiation of oxygen, 18 enriched water, through the 18O-PN-18F reaction. Carbon-11, with a significantly shorter half-life of approximately 20.4 minutes, is generated through reactions such as 14N-P-alpha, 11C, and requires rapid synthesis and immediate use due to its fast decay. Other short-lived isotopes include nitrogen-13 and oxygen-15, with half-lives of approximately 10 minutes and 2 minutes, respectively, which are primarily used in specialized physiological studies such as myocardial perfusion and cerebral blood flow imaging. Gallium-68, with a half-life of about 67.7 minutes, is obtained from germanium-68/gallium-68 generator systems, offering a practical alternative to cyclotron-based production. In contrast, zirconium-89 possesses a relatively long half-life of approximately 3.3 days, making it suitable for imaging slow biological processes, such as the biodistribution of monoclonal antibodies in immuno-PET applications. The selection of a PET isotope is governed by its half-life, decay energy, and compatibility with the biological half-life of the targeting molecule. Short-lived isotopes are advantageous for minimizing radiation exposure, but impose stringent time constraints on synthesis and imaging, whereas longer-lived isotopes enable extended imaging protocols at the expense of increased radiation dose. PET radiopharmaceuticals, organic molecules and applications. PET radiopharmaceuticals are designed by integrating positron-emitting isotopes into organic molecules that participate in or mimic biological processes. Among these, fluorine-18 labeled compounds are the most extensively utilized due to the favorable chemical properties of the carbon-fluorine bond, which imparts metabolic stability and allows for diverse synthetic transformations. The most prominent example is 18F-fluorodeoxyglucose, a glucose analog that accumulates in cells with high glycolytic activity, making it invaluable in oncology for tumor detection and staging. Other fluorine-18 labeled compounds include 18F-fluorothymidine, which serves as a marker of cellular proliferation, and 18F-fluorodopa, widely used in neuroimaging to assess dopaminergic function. Steroid-based tracers, such as 18F-fluoroestradiol, enable the visualization of hormone receptor expression in cancers, particularly breast tumors. Carbon-11 labeled compounds offer a unique advantage in that the radioactive isotope can replace stable carbon atoms within a molecule without altering its biochemical behavior. This property allows for the development of tracers that closely resemble endogenous substrates. Notable examples include 11-C-raclopride for imaging dopamine receptors, 11-C-Pittsburgh compound B for detecting amyloid plaques in Alzheimer's disease, and 11-C-choline and 11-C-acetate for imaging tumor metabolism and proliferation. Radiometal-based PET tracers, although not strictly organic in structure, are frequently conjugated to peptides or biomolecules that confer biological specificity. Gallium-68 labeled compounds, such as dotatate and PSMA targeting ligands, have gained widespread clinical use in neuroendocrine and prostate cancers, respectively. Similarly, zirconium-89 labeled monoclonal antibodies are employed in immuno-PET to track the distribution and target engagement of therapeutic antibodies. Synthetic strategies for PET radiopharmaceuticals. The synthesis of PET radiopharmaceuticals requires rapid, efficient, and reproducible chemical transformations due to the short half-lives of the radionuclides involved. In the case of fluorine-18, nucleophilic substitution reactions are commonly employed, wherein 18F-fluoride displaces a suitable leaving group, such as a tosylate or mesylate, on a precursor molecule. This approach is widely used for the preparation of small molecule tracers. Electrophilic fluorination methods are less common, but can be useful for specific substrates. Advances in late-stage fluorination have enabled the incorporation of fluorine-18 into complex molecules without extensive precursor modification. Carbon-11 chemistry is dominated by methylation and carbonylation reactions using reactive intermediates such as carbon-11 methyl iodide, carbon-11 methyl triflate, and carbon-11 carbon monoxide. These reactions facilitate the incorporation of carbon-11 into a wide range of functional groups, including amines, alcohols, and carbonyl compounds. Due to the short half-life of carbon-11, these reactions must be performed rapidly and often in automated synthesis modules. For radiometal-based tracers, the synthesis typically involves coordination of the metal ion with a bifunctional chelator that is covalently attached to a targeting molecule. Chelators such as DOTA and NOTA provide stable complexes with gallium-68 and zirconium-89, enabling the development of peptide and antibody-based radiopharmaceuticals. Challenges in PET radiopharmaceutical development. The development of PET radiopharmaceuticals presents several significant challenges. One of the primary limitations arises from the short half-lives of many PET isotopes, which necessitate rapid synthesis, purification, and administration. This requirement imposes strict constraints on reaction kinetics and often demands the use of automated synthesis systems located in close proximity to imaging facilities. Radiochemical complexity also represents a major hurdle, particularly when incorporating radionuclides into structurally complex organic molecules. Achieving high radiochemical yields and purity within a limited time frame requires careful optimization of reaction conditions and precursor design. In addition, maintaining high molar activity is essential to avoid pharmacological effects and ensure that imaging reflects true biological processes rather than tracer-induced perturbations. Biological factors further complicate radiopharmaceutical development. Issues such as nonspecific binding, metabolic instability, and limited ability to cross biological barriers can adversely affect imaging performance. For central nervous system applications, the ability of a tracer to penetrate the blood-brain barrier is a critical determinant of its utility. Infrastructure requirements also pose practical challenges, as the production and use of PET radiopharmaceuticals require access to cyclotrons, specialized radiochemistry laboratories, and trained personnel. Furthermore, regulatory considerations related to radiation safety, sterility, and clinical efficacy must be addressed before a radiopharmaceutical can be approved for clinical use. In conclusion, PET imaging is a powerful and versatile technique that relies on the integration of nuclear physics, organic chemistry, and molecular biology. The development of PET isotopes and radiopharmaceuticals has enabled unprecedented insights into physiological and pathological processes. Fluorine-18 and carbon-11 remain the most widely used isotopes for organic tracer development, while radiometals such as gallium-68 and zirconium-89 have expanded the scope of targeted imaging. Despite challenges related to synthesis, logistics, and biological performance, ongoing advances in radiochemistry and molecular targeting continue to enhance the capabilities of PET imaging, supporting its growing role in precision medicine.